Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have a returning guest. His name is James Shapiro. He's worked as a professor of microbiology at the University of Chicago since 1973. Very, very long, distinguished career in microbiology, perhaps some evolutionary biology, things like that. He's a great person to speak to. He's got tons of knowledge. He has a recent book he's come out with is Evolution, A View from the 21st Century. He's written countless dozens of articles and papers and many books. So, James, thanks for coming. How are you doing?
I'm doing okay. It's a pleasure to be with you.
Yeah, I'm talking to you. I guess we can ask about this first in the time of the virus, the SARS-CoV-2. So, unfortunately, everyone's talking about it, but I would guess you have some unique insights as to not only this virus, but viruses and how they interact with the evolution of all things. So I guess you've written an article about it recently. What's your take on the current situation? And let's talk about viruses in general.
Well, first about COVID, a friend of mine found an article which claimed that COVID was genetically engineered by human beings. And we got some young people to do the sequence analysis and check that out. And it turns out not to be true. The same things that this person felt were genetically engineered were found in related coronaviruses found in bats and other animals. So that rules out human genetic engineering of the virus. That's one of those things that's been going around, but it turns out to be incorrect. And we looked at it very carefully.
Well, a quick question about that. So does that mean you didn't see any novel sequences in SARS-CoV-2? How would you know if it was engineered by humans, even if such a thing could be done?
Well, this person was picking up some homologies to HIV and other immunodeficiency viruses, but they're found in SARS virus isolated from bats. So nobody engineered that.
Right, okay.
And those just happen to be those kinds of sequence similarities which come up either by chance or probably in this case because the proteins that are being encoded do similar functions in the two viruses. Would
it be an amazing feat if people could engineer a virus, one that had directed properties? Is that, you think that's technically feasible, or is that a way off?
No, it's clearly technically feasible. And I remember when I was starting my career, we were doing this all the time with bacterial viruses, bacteriophages. So we would engineer them or isolate ones which had picked up pieces of the bacterial chromosome. And viruses in evolution play a lot of different roles. Most people think of them simply as parasites, but in fact, viruses do a lot of positive things, and they are capable of picking up DNA and transmitting it to cells and providing useful functions for cells. So some of the viruses in bacteria in nature, which are found help protect the bacteria from parasites, from protozoa and other amoebae and things that eat them up, and they produce toxins which are knock off these predators, and so it's useful to the bacteria. Unfortunately, when the bacteria tend to get into the human environment, in our water supply or something like that, they can cause disease, and the same toxins the bacteria use to protect themselves often contribute to human disease. So this is a property called lysogenic conversion, where a bacterium picks up a virus, incorporates it into its genome, and acquires new properties as a result of that.
Wait a minute. So what if I was infected by a bacteria, I don't know, E. coli of whatever strain was virulent but then I was infected by E. coli that also had a virus that was commensally living inside of it. I mean, does that happen? And then we would mistake and wonder why, for instance, an E. coli infection one time got me sick and another time it didn't get me sick, or it got these people sick but not these other people sick. Do you think this is a hidden determinant of what a bacteria, for instance, can do to us if it's pathogenic?
Well, it's not so hidden because it's been known about for about, it's going on to 75 years now.
I guess I'm not everybody. I don't know about it, and I've never heard it talked about. That's why I asked.
Yeah, well, I'm interested in viruses as to what they can contribute to cells.
Right,
and
that's better,
I think about viruses as kind of the R&D sector of the evolution process. It turns out that viruses have a lot of sequences which have never been seen anywhere else in nature. We don't have any idea of where they come from. But very frequently, viruses contribute sequences to living cells. But it's very rare that the viruses acquire sequences from cells. So it's kind of a one-way transfer system. And
is it rare? I mean, like for instance, I looked up GIS AID. They're cataloging the COVID-19 sequences, and they have 4,000 something, and they said there's already 1,600 variants. So where would the virus, how would it vary? Is it just, I don't know, is it RNA recombining and mutating on its own, or is it picking up things from human hosts and endogenizing it into the virus itself and changing that way?
Yeah, the coronaviruses are RNA viruses. And as far as I know, they don't exist in DNA form. So they don't become part of our genomes. There are other viruses like the retroviruses, which are actually copied into DNA and are a very important part of our genome. And some people would like to think that all they do is cause trouble. But it turns out that in evolution of vertebrates and evolution of mammals in particular, retroviruses have been extremely important. They allow us to have an immune system. They make it possible for pregnancy. They contribute to the development of the placenta. And
I've heard that,
So their viruses are playing a very positive role.
It seems like viruses are a vast library of, genetic ability that are all around us. And some we unintentionally harness, if they infect us, and some we try to intentionally harness. It's just weird. That's what it appears to me is this vast library of possibilities that is everywhere if we can understand its language and somehow use it.
Right. That's why I call it the R&D sector. It's where a lot of new things come in. And some of them prove to be very useful and very important, and others don't.
Do you think that viruses are alive, or at least contingently alive, once they enter into a cell?
Well, viruses are part of what we can call the biosphere. And they're really ways of cells communicating with each other. Viruses on their own can't reproduce or do much. But once they get into a cell, of course, then they can become active. And we focused on some of the pathological properties of the viruses. But as I said, they also do a lot of useful things for cells.
Well, do you think that viruses are adapting towards a goal, becoming less virulent, more commensal, or maybe even trying to endogenize into the host if they could? And I know most can't, but do you think viruses have a goal? Are they adapting towards something, or are they just randomly changing? And they don't seem to be. They seem to be have some kind of directed behavior of adaptation.
Well, each kind of virus is different, and each changes in its own way. It's generally not random change. But one pattern that we do see is that new viruses come in, and when they first come in, just as we see with COVID-19, they can cause a lot of trouble. But after a while, the host and the virus become adapted to each other. And then the host can make use of the virus. And this is certainly what happens with endogenous retroviruses. Those are the ones that are involved in the evolution of pregnancy and the immune system and other systems in our bodies.
Do you think that that adaptation is driven both by our own cells and by the virus, or just by our own cells?
Well, once the virus has become endogenous, that means it's part of the genome. It's really part of the cell system. And from that point on, it's the cells making use of the retrovirus for its own purposes.
Okay. Yeah, I just wondered again if we're doing the adapting, or the virus is doing the adapting, and I know a lot of viruses we may live with our entire lives, and they may not affect us, or they may only come out periodically, like herpes or HPV. Or it's weird that, and amazing to think that a lot of creatures will have viruses that are in their cells and hanging out, and they never leave, but they never seem to cause any trouble.
That's true. I mean, some genomes are a large fraction of them are viruses. In fact, our genomes, I think, are 14% retrovirus, which is a lot more than the part of the genome that codes for proteins.
That's amazing.
Yeah.
So what is what are the role of viruses in evolution then? Do you think that they're I don't know, are they guiding evolution? Like what, instead of like a tree of life, when you add viruses to the tree or whatever structure, what does it look like now to you?
Well, as I tried to indicate, viruses are a source of new information, which may be useful to evolving organisms at critical junctures in evolution. And they're vectors for transmitting information from one organism to another. And then when they become part of the genome, they provide extra potential for change in the genome. And I think the key thing to recognize is that all organisms have to evolve to survive. Evolution is not an accidental process. It's a necessary process because conditions are always changing. And viruses are part of the toolkit that living organisms have to evolve. And we should look at them that way, not just look at them as disease pathogens.
Yeah, I agree. If you look at a bacteria, does it seem to be able to use viruses? It seems to be able to use viruses as tools pretty readily, from what I've heard. It can snip out pieces of the virus and then put that into its own DNA so that it can recognize it and defend against it. I mean, it seems like there's a big dynamic there. But what about our somatic cells? Do you think that they have much ability to use viruses as tools?
Well, we have to explore that. In some cases, viruses can be activated in somatic cells, and they can change them.
Really? When would they be activated? How?
Well, one of the things I'm working on now with some colleagues is the relationships between cancer evolution and organismal evolution. And in cancer evolution, we can see very large-scale changes that occur as the cancer develops. Some cancers activate some of these endogenous viruses and undergo changes with the viruses. And
what do you mean they activated? Like so the endogenized bits of virus.
Right.
How do you know they're activated? Do they form virus particles again in the cell, or do they just act as genes and they're upregulated and they do things? Well,
some of the viruses are defective and they can't make particles, but others aren't. And typically in human cells, for example, the endogenous retroviruses are kept silent by methylation of the DNA. They become part of what's called heterochromatin. That's the part of the genome that's not expressed. But if the DNA is demethylated, then the viruses become active. And they can, when the retroviruses in particular, make DNA copies of themselves, which can then go to new places in the genome and encode new traits. And that's clearly happened in our evolution at important junctures. And we see that happening in some cancers as well.
Really?
Yeah.
What's the consequence of that? What happens when, the methylation is removed, the epigenetic marks are removed by in the cancer cells? What have we observed to have happened?
Well, we observe activity of these viruses, and that means increased genetic change. There are many other kinds of changes which can go on in cancer cells as well. And cancer is an evolution disease. It's cells evolving to grow in a way that they're not supposed to grow, and it's actually injurious to the host. And activating the retroviruses is part of that.
So it could, I guess to put it bluntly, if you have certain kind of cancer, could that cause a viral infection, but it's really coming from your own, coming from previously endogenized viruses. Could that, again, virally infect you, but it's all coming from your own cells?
That's a possibility, but it's actually the internal activity of the viruses, which is what we understand the best.
So what kinds of things have been observed when these endogenized virus genes are activated?
When you treat cancers with demethylating agents, you actually activate these retroviruses. Wow. And that can contribute to the formation of cancer cells, which are actually more virulent. But I don't know the precise details of that. What I do know, and what I think is interesting, is that we often find major changes in structure of the genome in cancer. It's what we call macroevolution, which is very different from the kind of microevolution that most people think about. And this is also what happens when new species or new kinds of organisms are evolve, that there's a restructuring of the genome and all kinds of changes occur. And what we've learned from cancer is that the same kinds of changes can occur in many different cancers, very complex changes that can occur at different places in the genome, but the patterns of change, the way that things change, are the same, meaning that they're established routines that the organism has, that the cells have for restructuring their genomes. And that, I think, is quite interesting. And it turns out that some of these changes that we observe, when we start to look for them in healthy tissue, we can find examples of changes that have occurred like that in healthy tissue. Because it's not that the cancer cells are doing anything new, they're just using a potential which is there within the organism to restructure its genome, and doing it because they're under some pressure to evolve. For example, there's a process called chromothripsis, which means chromosome shattering. And what you find is that only a single chromosome in the genome can be broken up into pieces, and the pieces can be rearranged and then put back together as a chromosome. Sometimes with duplication, sometimes with deletion, missing things. And this is very important, and this is characteristic of about, I think it's a quarter to a third of all cancers. And this can happen on different chromosomes. It can even happen in parts of a chromosome. And the sequences affected, or the chromosomes affected, can be different in each case. But this pattern is quite clear, and it's clearly the same process that's at work. So that our understanding of how cells restructure their genomes has been expanded to include these more complex rearrangements. And then if we ask what happens in historical evolutionary history, we find that chromosome changes and restructurings are an important part of that too. And that's what has been called macroevolution. Wow. It's very different from the small minor changes that make up microevolution.
I was thinking if the removal of methylation can cause, this restructuring, what happens when sperm and egg meet and they form a zygote? I had heard that most epigenetic marks are removed. I wonder if there's any viral activity or activation at that point, and if that actually helps to let the zygote differentiate and grow.
Well, I'm not an embryologist. But the one thing I do know is that cells of the body make little vesicles. And all organisms do this. They're all over the place, these little sacs of membrane which contain protein or RNA or DNA. And in the mouse, at least, and I'm sure this applies to human beings as well, some of the tissue in the male parent, the somatic tissue, has to produce these vesicles with certain RNAs in them and put them into the sperm for the sperm to fertilize an egg and lead to normal development. And what that says to me is that these RNAs are a kind of what we call epigenetic information, and they're influencing the way that the genome is programmed, if you will, to develop. And if they're not there, then development doesn't go in the proper sequence.
Yeah, extracellular vesicles, I know, are given off by all cells, and they're big time in cell-to-cell communication. And they're actually reminiscent of viruses. It's like RNA or other stuff in a little envelope that enters into other cells. And changes them genetically, at least epigenetically. So it
Right.
sounds very much like a virus. It's weird. It's like our cells have a virus type tool to affect other cells.
Well, yes, and it certainly negates the idea that somehow the germline is pristine and immune from the rest of the body.
Well, yeah.
Because the vesicles can do this. And I wrote about these vesicles and also about the viruses in a paper that I called No Genome is an Island. And I think one of the things we need to understand is how cells interact with each other and with outside sources like viruses in the course of evolution. And this is all over the biosphere, these sources of transfer and communication and variability. And I think the idea that an organism is fixed and has to stay that way is just not the right way to look at things. I think we have to look at life as a process of continual change. Just as you were saying, fertilize an egg, then you have to change the developmental pattern. And I was just in my discussions with a colleague who wrote a very interesting book, a woman named Azura Raza, wrote a book called The First Cell. I don't know if you've heard of it.
Yeah, I'm going to be speaking to her in a few weeks. I know she collected thousands of tissue samples.
Very interesting woman, and her book is very good. It's called The First Cell.
Yeah.
And she sent out an email to some of her colleagues about an article where they find that the methylation patterns, the way the DNA is methylated in cancer cells is different from those of ordinary cells, but it's more different from stem cells than it is from ordinary cells. And that's counter to the idea that somehow cancer is an atavistic kind of process where the cell goes back to the stem cell type and multiplies in an uncontrolled fashion. And what it tells us is that cancer cells are evolved and are even more programmed, if you will, than the normal somatic cells of the body.
What's if you look at cancer as an independent life form now that has its own goals and homeostatic drive and everything, with it inside of you, unfortunately, why is its goals seem to be destruction? Why isn't it commensalism or mutualism? Why does it not seem to be something that can persist in us forever?
So I think in cancer, what's happened is that a process has been under conditions where it normally wouldn't be activated, and it goes awry. It's out of control, and that's why it's so destructive.
Okay.
But the capabilities which are exemplified by the cancer cells to change themselves are capabilities which are used normally in evolution to change the organism when its environment changes dramatically.
So is cancer like a teenage superhero? He can't control his powers, but he has all these special powers and he destroys things in the meantime?
Well, that's one analogy you could use, I suppose.
I know it's a weird one, but how do you think cancer first arises? I mean, the classical theory supposedly is, magically, randomly, one cell mutates and off it goes from there. But I don't know. What are your thoughts?
Well, I don't know the answer to that question. It's not a random process because we know there are certain kinds of cancers which have a hereditary basis and are more likely in some people than others. So that's not random. We know that if you come in contact with certain things, you're more likely to develop cancer than if you're not. Like if you smoke, for example, you're more likely to develop lung cancer. So that's not random. So there are things which act on the cells of the body and induce them to undergo genetic change. And that's how cancer initiates. I don't think we've been able to look at the very early stages of cancer. One of the problems is that we've taken some laboratory models, and you can talk to Dr. Raza about this in more detail, of the earliest stages of cancer that we do in the laboratory. But cells in the laboratory are not like cells in the body. And so we can't take what we can do in the laboratory and use that as a model for what's occurring in the body. And one of the points of focusing on the relationships between organismal evolution and evolution of cancer cells is to see what kind of parallels can be drawn, and if it can help us get to a better understanding of the origins of cancer, and also to be able to detect cancer at earlier stages.
Yeah, it seems, this is my speculation, of course, but cancer is a forced maladaptation, when a group of cells is under persistent stress. They're continually adapting, but I guess their hand is forced in a way to adapt, and it's a maladaptation. From there, there's perhaps no turning back.
Yeah, well, there are all kinds of stresses that go on in the body, and we know in all organisms and all kinds of cells that stresses trigger some of these functions that the cells have to change their heredity, and that's obviously happening at the origin of cancers. And the more we can spare ourselves of triggering those events, the better off we are. So if on lung cancer, don't smoke. And that's turned out to be very effective in bringing down lung cancer. Other cancers, you don't want to be exposed to certain chemicals, and we can deal with that. Also, it's not clear that cancer is a single disease. By that, I mean that there are different kinds of cancers, and they may have different origins and different triggers.
Well, I've heard that some cancers are caused by viruses like HPV. What's the mechanism look like, and can we see it very early on when a virus appears to cause a cancer?
Well, the one that is understood perhaps the best is Rous sarcoma virus, where the virus has picked up a piece of the genome and expresses a protein, the SAR protein, unusually high degree, and that upsets the regulation of the cell and sets in motion a train of events in chickens that leads to the formation of sarcomas. I don't know about the details with HPV or with herpes viruses, but there are other retroviruses which have picked up so-called oncogenes and are infectious triggers for cancer. So that can definitely happen. Also, bacteria are triggers for cancer.
Then the microbiome itself, our microbial attachment is super important, and I know they have their own phage, but yeah, the microbial interactions with our own cells. I've asked before, some say yes, some don't know, but I would guess that our microbiome communicates with our somatic cells. They're certainly making metabolites for us, we're making stuff for them, but maybe they're using vesicles or plasmids to communicate and regulate our cells and vice versa. Who knows?
Yes, that's definitely going on. Actually, we're bringing our conversation almost full circle, because bacteria which cause cancer tend to make toxins which act on the DNA and damage it, and that destabilizes the repair processes. And then you get genetic changes and you induce a tumor. And those toxins are carried by viruses, phages, which infect those bacteria. And that's what gives them the ability to produce those toxins. So we see that the viruses carry these sequences for producing the toxins. In some environments where the bacteria are in, say, an aqueous environment, it's okay. It protects them from predators. But when they're in the human body, it can be a problem. So
I guess to return to a previous analogy, we can be, bitten by a mosquito that's carrying a virus that can make us sick. And I guess what you're saying is we can be infected by a bacteria that can make us sick, or the bacteria could be carrying a virus that could then make us sick, or a parasite that's carrying a virus. It seems like viruses, again, they can attach to just about any living thing and then cause disease and cause change on a massive scale.
Well, basically, that's correct.
That's amazing. Just as I'm saying it, I'm thinking, wow, this is crazy.
Well, that's life.
To me, it's amazing. I don't know that's a good. What amazes you? What amazes you about what you've been learning, or does everything amaze you?
Yeah, well, I would agree with that last statement. The more we learn, the more amazed we become. So we used to think that only kinds of changes which occurred in DNA or changes of nucleotides or loss of nucleotides creating frame shifts. That's what was the dogma when I was a graduate student in the mid-'60s. And then I had these mutations which didn't behave like nucleotide substitutions or frame shifts. And it turned out that they were pieces of DNA. And now I know that those are what are called mobile genetic elements or transposable elements, which move through the genome. And initially you might think, well, these are just, people would call them junk DNA and say that they were just harmful and were parasites. But now we know that this DNA is what allows evolutionary change to happen. And more than half of our genome is composed of this kind of DNA, this mobile DNA. And we're learning about how it's used at higher level regulation of how the genome functions. And what we're trying to understand with cancer and with organisms as well is how do they change? How do they use these mobile genetic elements and mobile DNA? And how do they create genomes which have different properties and generate organisms or cell cultures which can do different things?
So if I was to longitudinally sequence a whole bunch of people when they were little, then as they're middle-aged, and then older, would we see that their genes have changed? Their underlying DNA has rearranged and changed substantially?
I think so. I think there are actually some studies like that where they take different tissues and sequence them, and they find that there are DNA differences between the different tissues.
Wow. Huh.
I think what they looked at initially, at least, was what are called copy number variation.
And what's that?
Or CNVs.
Oh, what does that mean?
That means that the copy of a sequence, a particular sequence, a particular region of the genome changes. So you get duplications or triplications, or you get circular DNA, which can replicate independently. And we see these things happening in cancer, but we're finding out more and more that they're happening in normal tissue.
Weird.
And some of them may be part of the developmental program, if you will, of those tissues. I don't know how far advanced the analysis has gone. And I probably shouldn't say too much more because I'm not an expert in this area. But I think it's an interesting area to think about. The genome in every cell is not identical. And that makes sense because the cells are not identical.
Well, but supposedly they all have, you can induce pluripotency and then they can supposedly do everything. But I guess they're differentiated differently, but they may be different on a deeper level, a genetic level, sounds like.
Yes. I mean, they have the basic, what a friend of mine who's a cancer oncologist, cancer cytogeneticist calls the parts list. They have the parts list, that 1.5% or less of our genome that encodes the proteins, so they can make different kinds of cells, and they can undergo those changes. But how those different parts of the genome or different proteins are made and when they're made is different in different tissues. Also, what changes, of course, in the course of cancer. And I think that there's a lot that we still don't understand about how differentiation and morphogenesis are controlled.
That's right. Yeah. Why do 100+ billion people have one liver instead of two, and it's always a similar shape and a similar placement versus other organs? And yeah, it's weird. Where is that plan? What are there genes that control it? Like, how could you know that from just one cell differentiating in the right directions? And again, cells forming not only the right structures, but the right size of them and in proportion to each other at distance and everything. It's weird.
Right. So there's a lot there to learn. And we have all this molecular biology and we can read all these DNA sequences, but there's other things going on that we haven't figured out yet. And I think it's always a good idea to keep in mind that what we don't know is probably much greater than what we do know.
Yeah. How do we know what a particular gene will do? I guess the only way is we observe it and we see that, in all these creatures, this sequence seems to code for this protein or whatever. How would we understand the language of RNA or the language of DNA independently? So we could look at a sequence and say, oh yeah, based on the order of things, it's going to code for these things?
Well, I think it's a mistake to think about it independently. I think that's held back our understanding of how these systems work, because you've got a genome which can make all different kinds of cells. So it's not just the proteins that it can make, but it's how they're expressed and in what combinations. And they can make very different kinds of tissue. They can make teeth, they can make intestines, they can make livers, as you pointed out, hearts, eyes, brains, skin, all different kinds of tissues. And it's not because of the content of the genome, it's how the genome gets used. And what we don't understand is how the genome is I don't know how to put it, how the system is controlled during development so that we get these different kinds of tissues. And we also don't understand how these how different kinds of organisms form in evolution. But what we need to look at is not necessarily just the bits and pieces, the individual proteins, but we have to look at how the system is organized and how it accomplishes these remarkable feats of diversification and of utilization of the information in so many different ways.
Yeah, like the question of, where is the life in a cell, in a bacteria? Has anyone tried to, take a cell apart, selectively remove pieces of it until it's quote unquote dead? What makes a cell alive? What's the minimum assemblage of atoms or molecules to make something alive? It's weird.
Well, it's interesting because you're asking a kind of a little bit an origin of life type question. And I'm not very keen on those because I think we don't know enough to discuss that. But it's interesting that Craig Venter and his colleagues have made what they call synthetic organisms from mycoplasmas, which are the living cells with the smallest genomes. But in fact, all they've done is by chemically synthesizing sequences which have the same coding capacity as the natural sequence. And not all of those synthetic sequences are functional.
But some are.
We don't know how to make a living cell from the beginning. We can only do it by copying cells that already exist.
And we don't know what is it about that unique combination of elements that makes something living. Yeah.
I think that's correct. And it seems to be more than just the DNA sequence. You need the cell, you need the membrane, you need the metabolism, and you need the other features of the cell, which perhaps we don't understand yet.
Yeah. What do you sense that science is close to figuring out, perhaps in the next few years? Any major things that you're really looking to see when we can make a breakthrough on?
I think we're going to understand evolution in a different way. Certainly that's what I've been interested in. And also I think we're going to understand how cells operate in a different way. I was invited to contribute to a special issue of a journal on the subject of cognition. And I think I was invited because a number of years ago, I wrote a paper which the title began, "Bacteria are small but not stupid." And I was talking about all of the cognitive abilities that bacteria have. And I'm writing a paper now called "All Living Cells Are Cognitive," and trying to point out all the ways that cells pick up information from their environment, from other cells, about what goes on inside of them, so that they can repair themselves when things go wrong or there's damage inside of them. And I think we're going to think about cells in a more integrated, systemic, and cognitive way as we try and understand some of these mysteries that are there for us, because they're really problems about how systems change. If we think about development of different tissues and morphology, it's taking the genome as a database, and how does the system use it in different ways to make all of these different kinds of cells and tissues. So I think that's where progress lies. And I'm not exactly sure how we approach that. Some people use computer systems as a model, but I don't know that that's really the best model to use, because certainly living cells are far more efficient than any computer system.
Well, I don't think they're living machines. I think they are intelligent because the information they work with is ambiguous. There's nothing 100% specific, and they take contingent action, depending on what the information is that they're evaluating. And that's not a machine. That's intelligence that does that,
Well, where is the intelligence that's doing that?
I don't know, but it's there.
Right. Well, that may be something that we've got to figure out.
Yeah.
I don't know how close or how distant we are from that.
Well, I think the sad thing is that people aren't even allowed to challenge the assumptions, let's say, of neo-Darwinism. If you think about a problem and you assume different things instead of random mutation and natural selection, it opens your mind up to a whole different way of thinking. But very few people are willing to even do that. It's like they're afraid that they'll be ridiculed, and why not? Maybe that's where progress will come from, is by saying, all right, well, maybe these base assumptions are wrong. Let's assume this other way and see what happens. What if we assume a cell is cognitive in its own way? How does that change how we evaluate what it does and how it does it?
Well, that's exactly the point I'm trying to make. Yes, I agree with you.
Yeah.
And I think we need to change assumptions, especially when we have evidence to the contrary, that the basic assumptions are faulty. For example, the basic assumptions about random mutations in evolution, where we know that there are dedicated systems in cells for changing the DNA. So the basic assumption there is mistaken. And we have to understand that that's part of life. And in the problem of cancer, of course, is understanding how that triggering those systems can go awry and lead to cancer and death. But they're there, and they're important at other times in evolution. Yeah. So that's where we have to look at evolution, and it means probably looking at different parts of the genome that we're not accustomed to studying if you're using DNA as your starting point. And the point of writing about cells being cognitive, especially bacteria and archaea, the simplest cells we know about, is to point out that sensitivity and responsiveness and cognition are there in all cells. And if you start from that basis and ask, well, how does that cognition work? What is the basis of it? Right. How does it make things come out right? I think you perhaps will get different answers than if you say, well, if I understand all of the atoms in this structure of this protein, I'll know everything about it.
That's right. Yeah. I think that's neo-Darwinism or Darwinism was a great construct to help us understand things, but I think now it's become it's crippled science and thought, and I think it's become a poison that's excluded a lot of other thought. But that's just that's my opinion. So
right. Well, one of the reasons I got interested in cancer was that this colleague I was mentioning earlier, who was a cancer cytogeneticist, wrote a book called Genome Chaos, and he sent it to me. And it's a very interesting book, and it's a lot of the book is about how the idea that all that was important about genomes are the genes, rather than the structure of the genome and the organization of the genome, that this was misleading us. And so we're doing all of this sequencing, and we're probably sequencing the wrong parts of the genome, and we're not understanding what it is about the genome that is giving cancer cells their capacities.
Yeah, I don't know. Well, James, this has been a great call. We're out of time, but how can people find out more about your work, which is ongoing, and your papers, and how can they get their first exposure to you and learn more?
Well, they can follow my website, shapiro.bsd.uchicago.edu, and if you put that in, it will pull up my webpage, and there's a lot of information there. You can try and get a copy of my 2011 book on evolution. It's out of print now, but you can buy or even rent paperback copies of it. And I'm hoping to publish a second edition at some point with some of the more recent information in it, basically to show that the ideas about cells engineering their genomes and using the genomes as read-write memory systems have been supported by lots of new information that's come out. And that's where people can look. And also I had a blog on the Huffington Post for a while, and you can still access those if you go to James Shapiro or James A. Shapiro at Huffington Post.
Very good. Well, James, thanks again for coming back. It's always good to talk to you, and I appreciate it.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.